morning. My name is Mariano Alvarez. I will be kicking off the session. Talking about some work that we did to look at methylation variation that's induced across generations by a seasonal cue. So I'm just going to start by talking a little bit about the word epigenetics. I know it's a little bit of a noisy term, so I just want to give you sort of my operational definition for the purposes of this talk. So when I refer to epigenetics, I'm just going to be talking about mechanisms that change gene expression rather than concurrent change in DNA sequence. And since we've discovered epigenetic mechanisms, there's some evidence that these modifications show a degree of independence, particularly during inheritance from the underlying DNA sequence. And this has led to a lot of speculation that epigenetic mechanisms might be a novel unit of transgenerational information. And this speculation has led to a few hypothetical evolutionary consequences, some of which are a bit more extraordinary and some of which are a bit more ordinary. So on the extraordinary side, people have hypothesized that epigenetic variation is a non-random, environmentally induced source of phenotypic variation that selection can act on, and that the transmission of epigenetic states can actually enable a novel response to selection. And that's quite extraordinary. On perhaps more ordinary side or more classical side, epigenetic mechanisms have been hypothesized to be a mechanism of parental defense. But for either of these two hypothetical sets of hypothetical evolutionary consequences, they both really rely on the idea that epigenetic variation can be environmentally induced and transmitted with fidelity across generations. And so that's the question that we were really interested in our lab. So I'm going to focus the rest of the talk on just one epigenetic mechanism, DNA methylation. I know there's a few different types of DNA methylation. For this talk, I'm just going to be talking about 5-methylcytosine. And that's literally just a methyl group attached to the fifth carbon of the cytosine. It's the most well-studied epigenetic mechanism, and that's because it has a few interesting properties. The first is that DNA methylation is a regulatory mechanism that modulates gene expression, and sort of the canonical way that that's described is by blocking transcription when it exists in the promoter region. Actually, it seems like there's a few other ways that it affects gene expression, but this is just sort of an example. It's also environmentally responsive. It's actually been shown in a number of different species, but just in Arabidopsis alone, DNA methylation has been responsive to cold, to simulated pathogen stress, and to simulated herbivory. That's simulated herbivory right there. Okay, and perhaps most importantly, there's some evidence that DNA methylation might be transgenerationally stable, both across mitosis and across meiosis. What remains unclear is whether environmentally induced methylation is transmitted across the meiotic barrier. So that's what we were really interested in. That's what we'll be focusing on for this project. So our model to look at DNA methylation variation is Arabidopsis, and we're particularly interested in the relationship between Arabidopsis and vernalization, which is this transient seasonal cold cue. And vernalization is an interesting effect for a couple of reasons. Number one, it's ecologically relevant. It actually induces this life stage transition after the cue is relieved. Importantly, it's not stressful for Arabidopsis. They experience it just as a normal part of their lives in the wild. And we know from previous work in other labs and previous work in our lab that vernalization alters phenotypes both within generations by inducing flowering and across generations by changing germination proportions. So there's already this transgenerational component to vernalization. So it's a really good model for thinking about these transgenerational epigenetic effects. So we put all this together and designed a small project. We used one genotype of Arabidopsis to control for genetic variation. We generated vernalized and unvernalized samples, and we sampled immature fruits, which we'll refer to as the parents for the rest of this talk, and seeds, which will represent the progeny. We had 3 replicates per treatment generation, and we used whole genome bisulfite sequencing to look at DNA methylation variation in these samples, and we had a cutoff of 20x minimum depth. So I won't talk too much about our analysis methods, but if you are interested, I'm more than happy to talk about it after the talk. very briefly, we just identified contiguous covariant regions of DNA methylation. We scaled and quantified the average methylation frequencies within those regions, and then we used those as our regression coefficients to test for treatment effects just using regular linear modeling. And that linear modeling actually included post-hoc comparisons. So briefly, I just want to go over what expectations we might have. And actually, our expectation for this project was that DNA methylation has a genetic basis. Since we controlled for genotype in this study, we shouldn't actually see any transmission of environmentally induced methylation to the next generation. And that's shown all the way over there on the left. We would see a response in fruit samples, but not a response in the seed samples. Alternatively, we might observe stable transmission of DNA methylation phenotypes across the generations. And that would appear as a change in fruit and a concurrent change in the seeds in the same direction. The third thing we might observe is what we're tentatively calling cryptic transmission, and that's when we might see these sort of oppositional effects where the fruits respond in one direction and the seeds actually respond in a different direction, or when we don't see a response in the parent generation, but we do see a response in the progeny generation. And we're calling it cryptic because we think that this might be indicative of some kind of secondary mechanism that is actually the unit of information here, and that's being transmitted across generations and then re-inducing DNA methylation variation. But we're just unable to see it in our data because we only have methylation and sequence data, and so that's why it's cryptic to us. Okay, so what did our data look like? So first of all, right off the bat, we saw many more differences, by 2 orders of magnitude more differences between our 2 generations than we did in response to vernalization. So right off the bat, we're definitely looking at sort of a sparser genome-wide response to vernalization, but we did pick it up actually. So we had 91 methylated regions that responded to vernalization and 82 that responded to the interaction of vernalization and generation. So those are the sets that I'm going to focus on now, sort of zoom in on those. So 81 of those regions responded in fruits and not in seeds, and that's pretty indicative of no transmission to the next generation. 56 responded both in fruits and in seeds, and that could be either stable transmission in that they responded the same way both in fruits and in seeds, or that cryptic transmission where they could be responding oppositionally. We also found 24 regions that only responded in the seeds, and so that's pretty clear, I think, pretty clear evidence of our cryptic transmission possibility. So I know this is a really busy figure. I'll walk you through it very, very briefly, and then we'll zoom in a little bit. What we did here is we just plotted out the average methylation frequencies for each region on the top of the parents, on the bottom of the progeny. The main thing I just want you to take away from this is we actually saw a variety of different types of responses, both in the level of methylation and in the directionality from fruits to seeds. So this is just the regions that responded in seeds. So when we zoom in on some of these regions, we do see some evidence that's consistent with this direct inheritance possibility, where we see a correlation between the parents and the offspring. But actually much more often we saw evidence of this cryptic transmission, where we saw one direction in the parents and a different direction in the seeds, or no response in the parent generation and a response in the offspring generation. And actually, when we look at how many of each of those types we saw many more regions that display this sort of cryptic transmission version than this direct inheritance version. So where are these regions? What are the underlying sequences? Well, among all loci responding to vernalization, we saw primarily response in transposable elements. We did see some mobile RNAs. Those are RNAs that can actually make it out into the cytoplasm. 2 zinc finger proteins, which might be interesting because some zinc finger proteins interact with DNA methylation. But primarily, we saw changes in transposable elements. And actually, among loci that responded in seeds, and so these are the transgenerational ones, we actually saw only responses in seeds. Or sorry, in transposable elements. So that's pretty interesting, I think. So where does this leave us? Well, number one, we know now that non-stressful parental environments can actually induce transgenerational variation, DNA methylation, and that seems to be primarily in transposable elements. Interestingly, the response in the parents doesn't seem to predict the response in actually inheritance of the methylation phenotype itself. Okay, and so with that, I'm happy to take questions. I'd just like to thank Marta Robertson, the entire Donahue Lab, my PhD advisor, Christina Richards, and of course our funding source, and I'll take your questions.
Just have two small questions. The first one, have you differentiated the methylation in the genomic context, which means is it CpG, CHH, or CHG?
We have. I haven't showed that data. We see primarily CHH methylation, but we still have to go through and quantify that. But just sort of from my recollection, it's mostly CHH.
The second one is for the environmental induced transgenerational epigenetic variation can happen in two possibilities. The first one is the methylation can resist reprogramming or resetting during the meiosis. The second one is the same environmental cues can induce the similar methylation patterns in two generations. Which of the possibilities in your case?
Right. So I think it's probably not that it's resisting resetting, mostly because we saw a lot of this cryptic variation. It could be that the environment is re-inducing, but actually in our vernalization cue, by the time the seeds are actually developing, the vernalization cue is relieved. So the seeds are developing in the same environment in the two treatments. It's just a temporary cold stimulus that's actually inducing these changes. But of course, there is tissue that still experienced